Welcome back to Pharmaceutical Biotech, where we explore the fascinating world of genetics and molecular biology. Today, we have an exciting topic lined up for you: Sanger sequencing. Have you ever wondered how scientists decode the genetic information found in DNA? Well, that's exactly what we're going to delve into today. So, if you're ready to unlock the secrets of DNA sequencing, let's get started. First things first, what is Sanger sequencing? Well, it's a groundbreaking technique that revolutionized the field of molecular biology. Developed by Frederick Sanger in the 1970s, this method allows us to read the sequence of DNA bases, which is like reading the genetic instruction manual of life. But before we dive into Sanger sequencing, let's quickly review some basics. DNA, or deoxyribonucleic acid, is the molecule that carries the genetic information of all living organisms. It's made up of 4 building blocks called nucleotides: adenine, thymine, cytosine, and guanine, often abbreviated as A, T, C, and G. Now, you might wonder, why do we need to sequence DNA? Well, understanding the order of these nucleotides in DNA is crucial for a wide range of applications, from studying genetics and heredity to diagnosing genetic diseases and even solving crimes. Sanger sequencing is one of the earliest and most reliable methods for accomplishing this. So, how does Sanger sequencing work? It's a multi-step process, but I'll break it down into the key components. We start with a small piece of DNA that we want to sequence. We need to make many copies of this DNA, a process known as DNA replication. Next, we set up 4 separate reactions, each containing our replicated DNA, along with a primer, DNA polymerase, and a mix of A, T, C, or G nucleotides. Here's the twist: in each reaction, we add a small amount of a special version of these nucleotides, the dideoxynucleotides, or ddNTPs. As DNA polymerase tries to extend the DNA strand, it randomly incorporates either a regular nucleotide or a ddNTP. When a ddNTP gets added, it terminates the DNA strand because it lacks the necessary chemical group to continue the chain. After these reactions, we end up with a mixture of DNA fragments of varying lengths, each terminating with a ddNTP. Now, we need to separate these fragments based on size. We use a technique called gel electrophoresis, which involves loading our DNA fragments onto a gel, applying an electric field, and watching as the fragments move through the gel based on their size. Smaller fragments travel faster, while larger ones lag behind. Finally, we analyze the gel and read the sequence of the DNA. We start with the smallest fragments, which correspond to the end of the sequence, and work our way up to the largest fragments, revealing the complete DNA sequence. Now, let's talk about the advantages and limitations of Sanger sequencing. One of its key advantages is accuracy, making it a gold standard for validating DNA sequences. However, it can be time-consuming and is best suited for relatively short DNA fragments. In recent years, newer, high-throughput sequencing technologies like Illumina sequencing and nanopore sequencing have surpassed Sanger sequencing in terms of speed and capacity. However, Sanger sequencing remains valuable for specific applications. And there you have it, a brief overview of Sanger sequencing, a powerful tool that has shaped the field of genetics for decades. Whether you're exploring your ancestry or deciphering the genetic basis of a disease, understanding how Sanger sequencing works is essential. If you found this video informative, don't forget to hit that like button and subscribe for more exciting content on genetics and molecular biology. Thanks for joining us today, and until next time, keep exploring the wonders of science.